Hello and welcome to our final session on gene regulation. So let us briefly compare regulation of transcriptional activity between bacterial or prokaryotic cells and human or eukaryotic cells. In both cases, RNA polymerase is involved. However, in eukaryotes, there are three RNA polymerases, and the RNA which is in charge of producing proteins is RNA polymerase 2. In contrast, in bacteria, there is only one RNA polymerase, which uses the sigma subunit to bind to a promoter. Promoter do play a regulatory role in both cases, because promoters are actually the start portion of of a gene. Quite important difference between prokaryotic and eukaryotic gene transcription is that the ground state in prokaryotes is actually on. In contrast, in eukaryotes, the ground state is off. So in eukaryotes, repressor proteins are used to negatively influence transcription of a gene. However, even in bacteria, activation of a gene can be positive or negative. This is also the case in eukaryotes. Transcription itself and regulation is much more complex in eukaryotes. In order to allow RNA polymerase to produce a gene product, this actually requires the Tata box, promoter sequence, several enhancer or silencer structures, and transcription factors. Regarding transcription factors, there are two general types of transcription factors. The general transcription factors, for example, TF2D is an example I showed you before. And we have plenty of different promoter-specific transcription factors. In order to conduct transcription, general transcription factors are binding to the Tata box. This recruits promoter-specific transcription factors that together facilitate RNA polymerases binding to the DNA sequence, and transcription can start. Again, the ground state in prokaryotes is on, in eukaryotes is off. As I mentioned in the learning outcomes, you need to be aware of the fact that transcription factors can be very, very promising drug targets. Why? Transcription factors are very, very good drug candidates, not just in cancer, but in other disorders, because they are regulating plenty of genes. Each of those promoter-specific transcription factor is in charge of positively or negatively affecting the levels of expression of multiple genes. An example of a promoter-specific transcription factor is transcription factor nuclear factor kappa B. It is found in almost all animal cell types, and it can be activated by many, many different signals. It can be activated by stress, it can be activated by cytokines, free radicals, ultraviolet light, or bacterial or viral infection. On the right-hand side, you can see DNA with two NF-kappa B subunits tightly bound to the enhancer sequence they are supposed to bind. Importantly, NF-kappa B is constitutively active in more than 90% of all cancer types. In contrast, in healthy cells, its expression is very, very tightly regulated. Moreover, NF-kappa B is known to regulate at least 800 of genes in human cells, and this makes NF-kappa B quite promising drug targets. It is very, very, very promising, and indeed many different drugs are known to affect NF-kappa B activity. For example, leptomycin B is a very well-known NF-kappa B inhibitor. Methyltransferase inhibitors also downregulate the level of NF-kappa B activity. Many natural products are known to do the same, including curcumin and so on. So, how can gene regulation be achieved at chromatin level? We had a look at gene regulation at the level of transcription. Again, as we have learned in one of our previous lectures on euchromatin and heterochromatin, you can actually imagine fairly easy that making the DNA denser can affect transcription of certain genes in a negative manner. And an important event in preparing a gene for transcription is chromatin remodeling. This process is basically opening up the DNA. During chromatin remodeling, DNA or histones undergo acetylation and methylation is usually removed. Right? So we have open chromatin that can be accessed not just by RNA polymerase but also by generic transcription factors. So again, methylated DNA is condensed and acetylated DNA can be accessed. So usually during opening up of the DNA, methyl groups are removed by an enzyme called DNA methyltransferase and acetylation is added. This process is actually quite complex, and all you need to take home from here is to remember that gene regulation at chromatin level is achieved by chromatin remodeling, and you also need to remember that this involves adding acetylation to the histones and removing methylation patterns. Another level of gene regulation is by modification of mRNA, and RNA interference is actually a very important pathway in human cells that is used in different organisms to regulate gene expression. During RNA interference, double-stranded RNA molecules are produced in the cell, and this is actually fairly unusual because in our normal cells, single-stranded RNA is the norm. If our cell senses double-stranded RNA, this usually results in cleavage of double-stranded mRNA. So if siRNA, a short RNA molecule, binds to the mRNA, this produces short double-stranded regions on the mRNA. As a result, certain enzymes in our cells cleave those complexes and remove the mRNA. If the mRNA is removed, gene can no longer be translated into proteins and the expression level is actually reduced. Importantly, RNA interference has been recognized as a therapeutic mechanism because nowadays we can introduce short single-stranded RNA sequences into the cell. If we design those RNA sequences to bind to very specific RNA mRNA molecules that code, for example, for genes in charge of proliferation, we could selectively inhibit growth of cancer or tumor cells. So let us summarize what the levels of gene regulation in human cells are. And this is actually something you need to remember for the exam. So gene regulation can be achieved by alteration of the gene structure, right? So heterochromatin can be turned into euchromatin and vice versa. The next level of gene regulation is transcriptional control. And this decides whether RNA is created or not. And this is achieved by promoter-specific transcription factors that are expressed depending on the environmental situation, age, and so on. Additional level of RNA of gene expression regulation is processing of mRNA. We can have post-transcriptional modification, we can have processing of introns and exons, and last but not least, stability of RNA can be affected. And this is, among others, achieved by short interfering RNAs or RNA interference. Lastly, gene regulation can be regulated at translational level. There are certain modified proteins that can actually stop translation, but this is just additional information that you need to remember, and you don't need to remember the mechanism. With this, I would like to close our gene regulation session, and thank you very much for your attention.